Good afternoon, everybody. Welcome to the BMES Professional Development Webinar organized by the BMES Advanced Biomanufacturing Special Interest Groups. And the topic for today's webinar is tissue biofabrication. I'm Kai-Ming Yi, and I will be your moderator today. I'm also the chair and the professor in the Department of Biomedical Engineering and the Binghamton University, and a part of the SUNY campus is the State University of New York. So here's my email address. If you have a question, you can continue to send email to me. This webinar is organized by the BMES Advanced Biomanufacturing Special Interest Groups. The goal is to inform the emerging field of the advanced biomanufacturing. As you know, the cell manufacturing and tissue biofabrication pose a significant challenge to the future progress of the biomanufacturing. I hope this webinar will give you some general ideas about the field and spur more collaborations between academia and industry. After I go over a few housekeeping items, and I will tell you more about the 6 current and planned activities. Let me see. Before I begin, there are a few housekeeping items. And first, this webinar is being recorded for your convenience. The slides and archive the recording will be available in the next several days and through the BMES website. And any time during this webinar, you may submit the questions by using the online chat function to the lower left of your screen. Please submit your questions as we go, and your questions will be addressed by the speakers at the end of the webinar. So lastly, and please do not exit webinar right at the end of the webinar because there will be a brief evaluation. We would greatly appreciate your feedback, so please take a moment to complete the survey. Now, here's some polling questions we want you to answer in the next 30 to 40 seconds. I'm going to read those polling questions. Which of the following best describe you? Undergraduate students, graduate students, postdoc fellow, professor, industry professions, clinician, government professional, others. Please complete on those polling questions. Okay, so we're going to close the polling session right now. And now I'm going to provide you a brief overview about the Advanced Biomanufacturing SIG. The mission of these special interest groups is to bring the academia and industry together to promote the development of advanced biomanufacturing, as to foster the collaborations among investigators who are working or interested in the field. And lastly, we want to create a new model of educating and training the next generation of the workforce for advanced biomanufacturing. The SIG is particularly interested in these topics, and including molecular biomanufacturing, cell manufacturing, tissue and organ biofabrications, and also include enabling technologies for advanced cell manufacturing and tissue biofabrications, multi-scale computational modeling and simulation. The SIG is also interested in developing the standards and regulatory science in advanced biomanufacturing and supply chains, of course, we are more interested in developing the new models for training and educating the new generation of the workforce for advanced biomanufacturing. These are the activities SIG plans in order to achieve our goal. And the SIG organizes annual Advanced Biomanufacturing Special Interest Group meeting, and a meeting usually in August every year. We also organize the special sessions in the BMES annual meeting. SIG also awards the Advanced Biomanufacturing Junior Investigator Travel Award at the SIG annual meeting to recognize their contribution and their achievements in the field. We also award the postdoctoral and graduate student travel award at the annual meeting to recognize their creative work in the field. SIG also publishes special issues on advanced biomanufacturing in peer-reviewed journals. We also organize the position papers in advanced biomanufacturing. Here's the overview of the SIG activities in 2016 to 2017. And we currently have 172 members. In the last year, in 2017, BMES annual meeting, so we added 2 sessions, include on cell manufacturing and tissue biofabrications. So we published the special issues in ACS, Biomaterial Science and Engineering. So we held the inaugural 2017 annual meeting in August 2017 at the University of Wisconsin in Madison. In 2018, we plan to publish position papers on cell manufacturing and tissue biofabrications. We also plan to have a second annual meeting in August 2018. So finally, we plan to establish a cell manufacturing and tissue biofabrication track in 2018 BMES annual meeting. So here's the information and show you how you can join the Advanced Biomanufacturing Special Interest Groups. And please visit the website and sign on for the special interest groups. So this is the editorial notes and the special interest group SIG wrote for the special issue published in the ACS Biomaterial Science Engineering in May 2018. And this is the advertisement page for the first SIG meeting, and that was held in the University of Wisconsin in August 2017. And these are the sponsors helped and launched the meeting. They included the NSF, LICA, and also University of Wisconsin WPI, and also White Cells. These are the committee members helped organize the meeting and develop a successful program for the meeting. And these are the speakers, and included the speakers from industry, from GE, Lonza, and also from academia. So now I'm going to introduce today's speaker, and Dr. Bill Murphy, and he is the co-director of the Stem Cell and Regenerative Medicine Center at the University of Wisconsin in Madison. So Dr. Murphy is a Harvey Spinger Professor of Biomedical Engineering. He is also Professor of Orthopedic and Rehabilitation. So he is also the Director of the Human Map Center at the University of Wisconsin. His research interests focus on creating new biomaterial inspired by the materials found in nature, and his research group is using the new materials to understand the stem cell behavior and to induce the tissue regenerations. He has published more than 150 scientific manuscripts, filed over 40 patents, co-founded multiple startup companies, and received awards that include the National Science Foundation Career Award and Wisconsin Villas Associate Award, and also the H.I. Romas Fellowship. He was inducted as a fellow in the American Institute for Medical and Biological Engineering. Our second speaker is Dr. Guohao Dai. He is currently associate professor in the Department of Bioengineering at Northeastern University. He received his BS and bachelor degree and master degree in mechanical engineering from Peking University, and his PhD degree in biomedical engineering from MIT's HST program. He completed his postdoctoral training in the Division of Health. He completed his postdoctoral training in the vascular biology at Harvard Medical School. His current research interests focus on the 3D bioprinting, stem cells, and vascular bioengineering. His research are continuously funded by many agencies, including NSF, NIH, American Heart Associations. Dr. Dai received the Scientist Development Award from the American Heart Association, also the Faculty Early Career Award from the National Science Foundation. He also received the Rising Star Award from the Biomedical Engineering Society and Institute of Faculty Career Award. I would now like to welcome our first featured speaker, Dr. Bill Murphy.
Thank you, Kai-Ming, and it's a pleasure to have a chance to talk to everyone who's joining the webinar today about some of our work in this area. So what I'd like to talk about is the engineered assembly of stem cell-derived human tissues for multiple purposes, but emphasizing the use of these tissues for discovery and therapy. I'll start just by disclosing a couple of relevant financial relationships. So the concept of self-organization or self-assembly of human tissues has been quite popular in the scientific literature and the engineering literature over the last 5 years. And the schematic at the top of this slide is from a Nature Methods news article from a few years back, describing in very general terms the concept of self-organization. The concept is that under the right circumstances, precursor cells, stem cell-derived cells in particular, have the potential to assemble into aggregates that become three-dimensional and ultimately take on some of the structural and functional characteristics of human tissues and organs. And there are some high-profile examples of this that have been published, including development of optic cup tissue, brain organoids, liver buds, and now several other examples in the scientific literature. Obviously, these kinds of human tissues on a chip or in a dish can be quite useful as models for human biology and as tools for drug discovery and for toxicology screening. But in order to be useful in those kinds of contexts, there's a real need to enhance the engineering parameters or improve the engineering parameters associated with the use of these tissues. So we've become interested in addressing a few of the critical challenges associated with these tissues in a dish. The first is robust and reproducible. So essentially making these kinds of tissues such that they have the same properties from well to well or from experiment to experiment. The second challenge is the ability to generate quantitative outputs, which oftentimes with complex multicellular tissues is very challenging. And then finally, one particularly important parameter is to make these systems transferable to pharmaceutical environments and toxicology screening environments so that they don't require very highly specialized expertise and very high-tech engineering methods that would be difficult to reproduce in different environments. So we very much are interested in creating solutions that an end user can access, whether they're at a pharmaceutical company, in an academic lab, in a government lab, or elsewhere. Our particular interest is in using biomaterials as a tool, and the concept is essentially to use materials as a way of giving instructions to cells as they organize into tissues. And what you're seeing here is from a review paper published a few years back describing some of the material properties that can influence stem cell behavior. every properties ranging from simple mechanical stiffness to much more complex binding affinity of materials for cell-secreted growth factors, can significantly influence stem cell behavior. And we and several other groups over the last decade have tried to identify and study the effects of materials on stem cell behaviors ranging from attachment and proliferation to differentiation and even tissue assembly. So we've become particularly interested in developing synthetic materials that have properties that will encourage stem cell-derived cells to assemble into stem cell-derived human tissues in a dish. As you're seeing from this slide, there are several different categories of parameters that can be quite important in dictating stem cell behavior. There are several listed here that are present in essentially every cell culture that one would generate. Nanotopography, stiffness, degradation byproducts, simple chemical functionality, molecular flexibility, cell adhesivity, and binding affinity. And if we consider even this limited parameter space, there are already many, many, many hundreds or even thousands of different materials that could be generated that have different levels of these different parameters or different types of embodiments of these parameters. So we became interested very early on in screening synthetic materials to identify unique customized materials that would promote specific cell behaviors. So I'll describe that a little bit on the next slide. Oh, sorry. So as early as 2008, my lab began screening individual hydrogels and other synthetic substrates in an effort to control stem cell behavior. And the concept is that if we have a rare behavior that stem cells are capable of undertaking, say expansion of a particular stem cell population or assembly of that population into a tissue, we can array out hundreds of different materials, seed the cell type of interest onto or into those materials, and simply observe whether those cells have undertaken the behavior that we're interested in. And so we've published a series of papers on this concept, and the movie that you just saw, that I'll play again, describes one of our array-based methods, essentially the ability to array out different materials and characterize how they influence stem cell properties. So we can now create hundreds or even thousands of different materials and try to zero in on materials that encourage self-organization of stem cells into human tissues. Okay. The other component that we need in these strategies is the cell population. And what we've focused on is using stem cell-derived cells. So we start with human pluripotent stem cells, typically human-induced pluripotent stem cells. And we can differentiate these cells into progenitor cells, like neural progenitors, endothelial cell progenitors, mesendodermal cells that are similar to mesenchymal stem cells, and microglia precursors. We can combine these with synthetic materials and encourage assembly of a tissue. And what we find attractive about this approach is that we can derive all of the cell components from patient-derived cells, which means that we can model individual human variability as well as potential disease and disorder derived cells. And we can start with pure cell sources that come out of cryopreservation, combining those with synthetic materials. So this is an approach that minimizes the typical variability that's been seen in developing multicellular tissues. So this slide gives a couple of examples of what can be done using this kind of an approach. So if one has a pluripotent stem cell-derived astrocyte population on the lower left-hand side here, these can be seeded onto a synthetic material, and we can observe the cells while they attach and become activated. So tracking an individual cell population can be useful for simple cell-based assays. If we want to go up one level of complexity and look at organization of a tissue from an individual cell population, we can look, for example, at pluripotent stem cell-derived vasculature. So this is a movie of endothelial cells attaching to a surface and undergoing assembly into a network of endothelium, which can be a useful screening outcome that relates to angiogenesis. And then finally, the perhaps highest level of complexity here involves assembly of human organoids in a dish by seeding pluripotent stem cells onto a substrate and encouraging their assembly from two-dimensional structures into three-dimensional tissues that are now forming, undergoing pseudomorphogenesis processes and actually forming into human organoids. So in each case, the approach involves seeding the cells into a synthetic environment and encouraging them to undergo what's been referred to as self-organization. Our goal in these cases is described on this slide. We'd like to be able to, on the lower left-hand side, use automated liquid handling robotics to create materials to be screened. So in essence, in step one, we're creating hundreds of materials, in this case in a 384-well plate. And once we identify the material that encourages assembly of a specific tissue type, we can then form that tissue type in every well of a 384-well plate and use it for automated high-throughput screening of potential drug candidates or potential human toxicants. So that's what you're seeing on the lower right-hand side here, a multi-channel automated liquid handling robot feeding potential drug candidates or potential toxicants to a human tissue. Okay. So that describes our two-step approach here, where step one involves identifying just the right material, what you might refer to as the Goldilocks material for assembly of a tissue on a chip. And the second step is to use those tissues in high-throughput screening, which creates a strategy that effectively is an off-the-shelf strategy. So what I'd like to do is describe a couple of different examples of this approach, one relatively simple one and one relatively complex one, to give you a sense for what where we're heading in this area. So the simple one is a vascular tubulogenesis screen. So one of the simpler tissue-based models that's been used in drug discovery and in disease modeling over the years has been this vascular tubulogenesis model. And in concept, what happens in this assay is that endothelial cells derived from human tissues are seeded onto matrigel, which is a mouse tumor-derived extracellular matrix that contains some 1,800 different human proteins, and they undergo assembly into a network of endothelial cells. And that assay has been quite useful over the years for identifying regulators of angiogenesis, but unfortunately hasn't been highly reproducible, due in large part to the inherent reproducibility in matrigel stocks and the variability in the use of matrigel from well to well and from experiment to experiment, maybe from batch to batch. So we set out to identify a synthetic material that would encourage assembly of endothelial cells into a network, but would be entirely synthetic and therefore more highly reproducible. So we did this in a paper that was just published a few weeks ago. What you're seeing at the upper left-hand side here are human iPS-derived endothelial cells assembling into a network on the surface of a synthetic hydrogel. These are hydrogels that are based on polyethylene glycol and have specific peptides incorporated within them to encourage assembly of these cells. And on the lower left-hand side, you can see what these networks look like for human umbilical vein endothelial cells and for human iPS-derived endothelial cells. These assemblies, when used as an assay platform to model angiogenesis, were much more effective than the matrigel-based platform based on Z-prime scores, and also were more accurate in identifying putative vascular disrupting compounds. These are toxicants that affect the human vasculature. So this is a promising approach, suggests that synthetic materials in some cases can outperform even some of the most complex extracellular matrices that are biologically derived. We've done similar kinds of studies in relatively simple neural screens. So we can use synthetic hydrogels to promote, on the top side here, differentiation of human pluripotent stem cells into neurons, and on the bottom side to create an assay that's based on iPS-derived neurons for human botulism neurotoxin. And in this case, the synthetic hydrogel-based approach outperformed the more commonly used laminin-coated plastic as a substrate for assembly of neuron cultures, essentially neuronal tissues on a chip. Finally, I'd like to introduce a more complex approach that we've been using to assemble human brain organoids on and within synthetic hydrogels. So what's happening in this case is that we're seeding neural progenitors, endothelial progenitors, microglia progenitors onto synthetic hydrogels. These hydrogels are designed to be remodeled by the cells to allow for assembly of human organoid. And what we found is that we can assemble human brain organoids that mimic critical aspects of early human brain development. So all of this is described in the published paper shown at the bottom of this slide. But briefly, what we found is that these brain organoid assemblies mimicked aspects of human cortex development. They were vascularized in 3 dimensions, and the blood vessels that are grown within these kinds of constructs mimicked critical aspects of human brain vasculature, including vessel wrapping by radial glia and the formation of end feet by radial glia on the endothelium. We also saw evidence of microglia-like cells that showed ramified morphologies and gene expression associated with microglia. These were highly reproducible, and in fact, these 3-dimensional multicellular tissues had reproducibility from well to well and from experiment to experiment that was equivalent to a typical monoculture of cells. So we were quite surprised by the level of robustness and reproducibility in these tissues, and that's all borne out in the 2 papers shown at the bottom of this slide. These also are amenable to high-throughput screening environments, so we can now generate these kinds of human brain organoids in 96-well plates, and we're moving now toward using them for high-throughput screening applications. Okay. We're building in now 3-dimensional vasculature into these systems. So here you're seeing a confocal micrograph of a 3-dimensional vascular tissue that's grown within a synthetic hydrogel and that we're now using to create vascularized human organoids on a chip. And our next step is to create fully perfusable and functional human organoids outside the body. This slide gives you a sense for what this might look like with a vascularized retinal organoid that we're working on in collaboration with David Gamm's group here at Wisconsin. So essentially we have a vascular bed that will feed a developing organoid and create a 3-dimensional perfusable vasculature. And you can see an example of this here, of retinal organoid that has CD31 positive endothelial cells engaged within it. And if I zoom in, you can actually see the individual organoid sitting on top of a vascular bed. Okay. So hopefully what I've described to you is that we have an approach to identify materials with unique properties. These materials create off-the-shelf systems for assembly of human tissues, and they really are designed to be transferable to pharmaceutical environments and to toxicology screening environments for use by many, many end users for a variety of applications, including for high-throughput screening. I'll finish just by introducing very quickly a couple of efforts that are ongoing at Wisconsin in this area, and that may relate to national efforts as well in biomanufacturing. So there's an Environmental Protection Agency funded center called the Human MAP Center here at Wisconsin, where we're developing human tissues on a chip for toxicant testing. And the idea is to create human systems that may ultimately replace animal model in understanding the effects of environmental chemicals on human tissues. We are partners with Georgia Tech, the University of Georgia, and University of Puerto Rico Mayagüez, among others, in establishing a new NSF Engineering Research Center for Cell Manufacturing Technologies called CMAT. And this uses, in some cases, biomaterials and other technologies to address bottlenecks in cell manufacturing, particularly for manufacturing of cell therapies. And then finally, we have a biomanufacturing effort we're developing here in the state of Wisconsin that's designed to really short-circuit the path from innovative technology development at technology readiness level 1 to 4, all the way through to technology readiness levels 4 through 9, so that we can push these innovations toward use in human patients and in a broad scope of end-user environments in pharmaceutical environments and in toxicology testing environments. And we have lots of engagement with industry as part of this. This is part of a technology roadmap that we were part of producing along with several partners, including Georgia Tech, and is available at cellmanufacturingusa.org, provides lots of information about what current industry bottlenecks exist and what opportunities there are, not just in R&D, but also in workforce development. I'd like to quickly acknowledge everyone who's been involved in this, a tremendous group of collaborators and efforts by some fantastic students, postdoc scientists in my group. A lot of this work led by Mike Schwartz and Bill Daly and Eric Nguyen, and involving several others here at the university. Thank you, and I'm looking forward to addressing questions as we get to the end of the webinar.
Thank you, Dr. Murphy. And now I would like to turn it over to our next featured speaker, Dr. Guohao Dai. Guohao, please.
Okay, thanks, Kai-Ming, for the introduction, for this great opportunity to have educational theory on the tissue biofabrication. So in the next 15 minutes, I would like to get an overview about 3D bioprinting. This is also now the enabling technology to fabricate the 3D viable tissue. So as we know, 3D printing is now the very enabling technology industry to be able to fabricate 3D materials, moving the material in the 3D and forming the 3D object. However, to moving this technology into the fabricated 3D viable tissue, there's a lot of technical challenging that involved with a lot of research in the 3D bioprinting field. So basically, industrial 3D printer frequently use harsh conditions. For example, they processing the material, metal or the plastic or the polymer using high temperature or very strong UV light or organic solvent, which make them unsuitable for the printing viable tissue with live cells. Right? So basically, the bioprinting is involved with processing biomaterial and cells under the gentle physiological conditions. So as similar to a printing, bioprinting basically involves the cell printing system and hydrogel material. So this is a mimic what we generally printing in our desktop printer. We need a printer, we need the ink, we need the paper, right? In this case, we need the cells and the printing system and the hydrogel. So in the next slide, I would like to introduce several general idea about the bioink, that is a substrate involved in the bioprinting. So because the bioprinting is a process involved with printing liquid solution and then allow the liquid solution to forming the 3D object. So there's a very unique property that we need to achieve is the phase changing, basically changing from liquid phase to the solid phase. So we call it the phase changing hydrogel. As we learn from a lot of approaches in the tissue engineering field, we can borrow those ideas and modify them for the bioprinting purpose. So here I list several examples of the biomaterial hydrogel. Their property can be manipulated and fit for the bioprinting process. So there are several mechanisms of causing the phase changing in the hydrogel. First is the temperature or pH. Basically, you change the temperature or change the pH of the hydrogel, and the hydrogel will change from liquid to the solid form. So, for example, the gelatin, agarose, Puronic, collagen, or matrix gel. These are the gel that are very sensitive to the pH or the temperature. There's also hydrogel that can be photo-cross-linked by involved with photoinitiator and the UV light. For example, the PEG hydrogel, methylated hyaluronic acid hydrogel, and the methylated gelatin hydrogel that can be UV cross-linkable, that can make them available for bioprinting purpose. The other type of using the ionic cross-linking mechanism, for example, in the alginate hydrogel with addition of calcium, and then the alginate can form a gel, or remove the calcium, then they can make them liquid again. Using enzyme can also make the chemical reaction that changing the hydrogel. For example, the fibrin can be made into a gel by adding the thrombin through the enzymatic reaction. So these are some of the examples of the hydrogel that can be used so during the bioprinting and fit into the bioprinting process. In the bioprinting, typically we also need to print a material that is as a supporting and eventually be removed. So this is called sacrificial material. Basically, sacrificial material is a material that will temporarily support the structure during the printing process, and then will be removed afterwards. So some of the material illustrated here can be modified by the temperature. For example, the gelatin, gelma, agarose, Puronic, or polysaccharide, for example, the sugar. I'll give you example later. The other is ionic, alginate. For example, you can adding a calcium or remove a calcium, make them change the phase, and then eventually be removed. So this material can also be made into the sacrificial material, into the bioprinting process. So next, I want to give you several examples of these bioink. So sacrificial material, this is an example of the two different type of sacrificial material as they respond to the change of temperature. So the first one is Puronic. So the second is gelatin or gelma. As we know, the gelatin is a material that if you heat it up from 0 degree to 37 degree, they will become liquefied. While this is a thermal responsive hydrogel that is liquefied by elevated temperature, the Puronic is acting as a polymer that acting in the opposite way. So Puronic, when you lower the temperature, and then it will liquefy. So showing here is the illustration of the shear module plotted here as function of temperature. So as you can see, when you lower the temperature, the Puronic will lower the shear module and become liquefied. Then it can be removed. Gelma on the other side, on the other hand, is when you elevate the temperature above 30 degree, and then it can become liquefied. So both of these can be used as sacrificial material, depending on the printing process. So besides those biomaterials that are frequently used, most of them come from nature or from synthetic. The normal tissue can also be decellularized and be processed and eventually modify their biophysical property and then make them suitable for bioprinting. So here is the example of using decellularized extracellular matrix as bioink. So showing here, Showing here is the actual tissue, and then being decellularized, and then this can be made into a powder, and then this powder can be made into the hydrogel format, and this hydrogel is very similar to the matrigel, and then can be changed into gelation format by changing ionic strength and also the temperature. So showing here several examples: cartilage tissue, hard tissue, adipose tissue, they all can be decellularized and made into the hydrogel, and can also be used bioink. So in this case, you can see the example of these type of tissue-derived bioink that will eventually be useful for biofabrication of the designated tissue type. So we have covered some of the bioink. So besides bioink, to do the bioprinting, we also need the hardware bioprinting, the bioprinter, and the printing technology. Basically, the technology to moving this material into the desired location. So next is example of the bioprinting techniques. So there's many different bioprinting techniques that can move the material from the reservoir to the design pattern. So, but here are three of the major category of the bioprinting techniques. So the first is called micro extrusion printer. So this is basically apply a pressure to move the shear thinning material. That is, the material has a shear thinning capacity that has a higher viscosity and it can be pushed out. And when the shear increase, the viscosity of the material decrease. And then this type of material can be easily put into the micro extrusion printer, can be pushed out through this type of printer. So this is suitable for some shear thinning material. The other is called inkjet printer, based on inkjet printing technology, by either using piezo actuator or microbubble technology, or using the electromechanical valve to generate a droplet of liquid. So this type of bioprinting technique is suitable for printing liquid biomaterial. And the third major type is called laser direct writing. So this is using a focused laser to move the biomaterial. So showing here, you can see a laser will be focused on the destination plane. The cell and the biomaterial is pre-prepared on a piece of ribbon. And then the focused laser will generate a very transient, very short pulse, localized heat. And then this will move this material, dropping this piece of material into the receiving substrate. And then the receiving substrate can be motorized under the computer-controlled XY stage. And then you can see a very precise pattern can be printed using the laser direct writing. There are also many other bioprinting techniques that are moving the biomaterial. So showing here is one of the examples of the bioprinting system. So basically, bioprinting system involved with hardware, and also a software interface to control the moving of this printing action and also the XYZ stage. So printing action illustrated here is the inkjet printing based technology. Is the cell and the hydrogel is loaded onto the syringe, and a gentle pressure is applied. So when the valve is open and closed under the control of the electromechanical valve, a small droplet can be generated and then print out from the dispenser. So next, I would like to show some example that illustrate the bioprinting, what type of structure that the bioprinting can generate. So the first example is a very simple example, using bioprinter to printing multiple layers of cells. So this is using bioprinting to printing layers of fibroblasts to mimic the dermis of the human skin, and also printing layers of keratinocyte to mimic the epidermis. So the bioprinted skin can be further maturated. Showing here is the bioprinted skin, multiple layer of cell gel structure can be further cultured and matured through several weeks of maturation culture, and eventually forming the tight epidermal junction. And also in the histological slide section, you can see they can be used to form histological feature that resemble human skin. So here's another example. Besides printing the cell and hydrogel separately, you can also mix the cell with hydrogel together and print them all together. Given if the cell and the hydrogel, they are compatible, and if action can be done under physiological condition. So this is example using printing mesenchymal stem cell mixed in the PEG hydrogel and simultaneously printing and UV cross-linking. So this action will make a very uniform cell gel mixture. Showing here is a bioprinted cell gel mix construct, and this construct can be further cultured under the culture condition. So this cell gel structure is under the differentiation, chondrogenic differentiation culture. So when the mesenchymal stem cell is under chondrogenic differentiation, they start to secrete glycosaminoglycan, the extracellular matrix molecule that are present in the cartilage. So next is example showing the usage of the sacrificial material. As we have discussed, the sacrificial material is a material that can be printed but eventually be removed just as a temporary supporting structure during the bioprinting process. So here, as you can see, we can printing a layer of collagen structure and then using a gelatin or cell gelatin as sacrificial material, printing a particular pattern. And then after the printing the sacrificial material and allow them to form in the gel, you can add in another layer, additional layer of the structure on top of it. So in this case, you can support multiple layers of cell gel structure. And showing here, after the bioprinting, the sacrificial material can be removed, allow to forming an empty hollowed channel. So this is a process you can use to printing a hollowed channel to mimic a vascular structure. So here's one of the examples that was done in my lab. So the hollowed channel was bioprinted, and then the endothelial cell was seeded simultaneously while the channel was printed. And eventually the sacrificial material was removed. And then this endothelial cell will cover the lumen of the channel, as showing here. Here is the cross-section of the channel, illustrated the confluent monolayer of endothelial cell cover the lumen of the channel, and this channel can be perfused. So showing here is the fluorescent microsphere to illustrate the lumen of the channel. So next is another example using sacrificial material. This is a very special sacrificial material. So this is a material that is called polysaccharide. Basically, is similar to the sugar that we are eating. So as you know, when you heat the sugar, it will melt. So this printer basically heat up the sugar at a very high temperature and then start to print out a filament. And then after the sugar is printed out, it quickly cool down and then forming a very brittle but yet quite strong structure. So here you can see using the bioprinter, you can printing a sugar lattice as a scaffold. So after this scaffold is printed, a hydrogel and cell mixture can be poured into the structure and then fill the 3D space. And then because this is the sacrificial material is the sugar, that can be easily dissolved when you're adding the water or the salt or the media to the 3D construct. And this approach is very versatile because it's just making the sacrificial 3D construct, and then any other cell gel mixture can be added and can be easily just added and poured into the structure. And this approach can make a quite large 3D tissue. So using Puronic as a sacrificial material.
Hey, Guo
this
Hao,
is
I'm sorry to interrupt you. Can you wrap up your presentation in 1 or 2 minutes?
Okay. So
yeah.
I will try to wrap up in a few minutes. So this is an example of in Dr. Jennifer Lewis' lab using Puronic as sacrificial material. As we see, the Puronic can be cooled down and then liquefied. So this is using sacrificial material to generate a vascular tubule in the kidney, and then the cell being seated on the surface of the tubule. And then they have showed this actually able to transport sodium potassium transport in a similar fashion as seen in the human kidney. So as we see, the bioprinting involves printing with material hydrogel that are frequently necessary for printing viable tissue. However, the hydrogel usually has very weak mechanical property. They usually cannot support a stronger, more stronger structure. So to approaching this problem, several other techniques to do the hybridized bioprinting. In this case, showing here is using printing a PCL as a scaffold that is very strong, and also printing the cell gel mixture to fill the space in between the PCL from the structure. That this hybrid structure has a very strong mechanical property, but also has the space for the cell gel fill the other part of the space, allow them to grow and remodel. So this hybrid structure showing here is to mimic a bioprinting of the human bone structure. And this structure was implanted into the mouse, showing they were able to generate a new bone and a bone marrow, showing here. Besides the bioprinting technology, there's also other techniques that people trying to modulate. For example, this using coaxial needle that you can not only printing a single line, but also printing a hollowed structure using biaxial needle. Another technique involved with patterned light. So this is using a patterned projected light apply on the surface of the UV cross-linkable hydrogel that it can generate a patterned vascularized structure. So basically, I just give some of the, there are many bioink development and also bioprinting techniques that we do not have time to go over. Basically, I have give you some simple example of the bioink and the printing techniques. So in summary, we can see the 3D bioprinting can construct 3D viable tissue using various hydrogel materials. There's a very strong need to develop new bioinks with improved biomechanical property and biological activity to match that of human tissue is critically needed. And also, vascularization remains an important challenge for long-term survival, remodeling, and maturation of live tissue. So I would like to acknowledge and thank the people in my lab, and also my collaborator during the past several years when I started to work on this project. So this will wrap up my part of the talk.
All right, thank you, Dr. Day. We would like to now take a time to get your questions and probably get one or two questions. But you may continue to submit your questions using the online charter features on your screen, and we will get to as many as we can. But also you can email all your questions to me and also the Dr. Murphy and Dr. Day, so we will get to you even after this webinar. So our first question is, what are the major benefits of the 3D bioprinting as compared to conventional tissue scaffolding? Guohao, do you want to answer the question?
Okay. So basically, there's a lot of technology we can learn from the conventional tissue engineering approach, also scaffolding. Typically, we can printing a 3D scaffold and then see the cell later. But the benefit of 3D printing is you can actually control the cell in a particular location. For example, if you want to printing a neurovascular junction, then you can printing an endothelial cell and the neural cell side by side at particular pattern. You can give them more precise control. While versus traditional tissue engineering approach, is basically mixing these two cells together, and it's hard to control their localization. The 3D printing gives you the benefit of control the localization in a 3D fashion. So that gives us a lot of enabling tools to ask a very interesting scientific question and also to better mimic the tissue architecture.
Thank you, Dr. Day. So the second question is, what are the major challenges in regulating tissue biofabricated products? Dr. Murphy.
Yeah, I think there are a variety of challenges. If the products are to be used and transplanted into human patients, then what we care about is safety and efficacy. And in the case of these 3D biofabricated products, there's a particular potential for these to replace the function of human cells that have been lost or damaged. And one of the challenges at the moment is that we don't have a standard by which to determine whether a particular batch or lot of these tissues is ready to be implanted into a human patient because we don't have very clear metrics for what would constitute a safe and effective product. So I think as we continue to develop these kinds of biofabricated products and as we continue to engage in clinical trials that explore their potential use in human patients, we need to continue to evolve our release criteria for these products such that we're identifying best practices for manufacturing tissue products, human tissue products that can be safe and effective, and we're identifying metrics during the manufacturing process that correlate to clinical success. It's difficult to do that at the moment because this is an evolving field, but very important for us to have standards coordinating committees that can help us to understand how parameters that can be evaluated during the manufacturing process might ultimately correlate to human clinical success. And some of the most recent work on CAR T therapy, I think, can be a real test bed in this area. What are effectively the CAR T manufacturing outcomes? What are the manufacturing parameters, metrics that have been measured during that manufacturing process that correlate with clinical safety and efficacy?
Thank you for the questions. And that is all the time we have for today. For those questions we did not get to, we will pass them on to the speakers to follow up with you. This will conclude our webinar. Thank you for joining us. Please stay online and take a few minutes to complete a brief webinar evaluation before you go. We would greatly appreciate your feedback. So we would also like to remind you the webinar is being recorded for your convenience. The slides and archived audio recording will be available on the BMES website. You will be notified by email when the archived material has been posted. We hope you will join us again for other BMES events. To learn about upcoming BMES activities and events, please visit www.bmes.org. Thank you.